gummy, yeah, wondering. Yeah. Okay. The placebo, if you like that too. And so we had to do that every time.
She was literally doing the placebo.
Oh, yeah.
Well, I just call it that.
That's great. Thank you. She was learning the lab. She had to just.
I see.
Okay. Oh, Brent, there's a woman who brought this down. Oh, I'm sorry. Oh, you got it.
Alright, so we're doing a test run tonight, but hopefully we'll be able to start
inserting a completely new gene in there.
Okay, so we just take the DNA stuff that you gave us last time, the Word file.
Yeah, but you're going to want a separate file. So you want the Word file with the Plasmid JE202.
Yeah.
You want a new DNA file for the message RNA for the accession number. You're going to want a separate file. So you want the Word file with the Plasmid JE202. You want a new DNA file for there's a little bit of feedback. Do you want to turn off the?
Yep.
So for those watching on Google Hangouts, we're doing the AF51312.1. This is actually a pretty accessible gene. It's from a grand fir from Northern California. So a nice quick drive. We could actually go copy this gene right from nature. And funny enough, this is one of the genes that kind of give you lemon flavor. I got these genes by doing a quick literature search on I guess it was, I did this last class, so I don't really remember where. But I finally found the papers on NCBI on PubMed. Now, if you don't have full access, I gave you guys some links of where you can find. Yeah, so they're talking to each other a little bit. Because when that moves, it turns off my computer screen. Anyway, sorry. So I found a paper. It's called "Engineering the Productivity of Recombinant Escherichia coli for Limiting Formation from Glycerol and Minimal Media." So we could probably grow this up also on regular LB. But this particular paper is doing it from glycerol and minimal media because that's probably some cheaper reagents for scale-up labor. This limonene could probably go for fragrance or even maybe for a food additive if it passes FDA. But that's where I got the genes from, and also pretty sure I got the accession numbers from those papers as well. If you cannot get the, clicking on the links and you don't have the full paper, I can't remember if I was able to get the full paper. We gave you some links here. Hashtag ICANN has PDF. I think that might work on Twitter. SciHub.io. Keep on searching. There's often different ways of reaching SciHub, but it's very reliable. You just put in the title of the paper and you'll actually get a PDF. It's super great. You can go on UCSF campus or some of the UCs, bring a dongle or a flash drive, and have the papers ready in hand that you want. You can search. They have licenses to all the papers, all the journals, or most of them. And so you can download them there for free as well. It's also great.
So anybody can just walk in.
Yeah, not all of them. It's not like Texas where you can go into any library. Sometimes you have to actually be an alum or part of the university. But I know the Doe Library here on the East Bay side allows everybody in. But their computers don't let you search, what is it called?
The article database, the UCSF does allow you to have articles.
Oh, you can search it, but it doesn't allow you to look at commercial sites using their computers. And I think to log on to their Wi-Fi, you have to be a student. So you can go in and use their computers for free. That's why you have to bring a dongle, and you can always, use your phone to find those papers if you're not quite prepared. And this may be true in other parts of the world. Like I said, University of Texas campus in Austin totally allowed anybody to come in and use computers for a set period of time, for like an hour. You can also try our mailing list, or even actually very reliably, email the first author. They'll often send you the paper for free and just happy somebody's reading it. So also it's a great resource if you have questions about what's going on in a paper. They're often available to answer those questions, but sometimes you have to ping them a couple times. Some of these people are PIs in labs, and so they're quite busy. All right, so I put these genes in there, so if you have some time, please go ahead and download those genes to your computer. I'm going to leave them on the screen right here so you guys can take that time. You can download these genes by going to NCBI, and then the search, go. In the search bar, right here, you simply just put that accession number in and push search, and lo and behold, when your internet works, ta-da, this is exactly the gene that we're looking for. And it's a reasonable size too, as well. But I want to remind everybody this gene is from a grand fir, and grand firs are eukaryotes. Pretty much, actually, all plants are eukaryotes. So does anybody remember the difference between a eukaryote and a prokaryote is in terms of coding sequence? Or sequencing, like their DNA sequence, not coding sequence. There shouldn't be too much difference. What was that?
Instead of being in the nucleus?
Yeah, there is no enveloped membranes, right? So that's one of the main, so the nucleus is an enveloped membrane, or enveloped organelle, sorry. But that's not exactly what I was looking for, but it does definitely plays a role in why the sequence is the way it is. For those who just came in, you might want to pull out a computer. We are downloading this gene here, the Apis grandis, which happens to be from a Northern California fir. Totally accessible. We can totally copy that gene out with some primers. We kind of went over that process last time. We'll do it again, a refresh this class. Yeah, no, it's right here. Click here.
Yeah. Oh.
Click on the link.
I have to click on it.
Yeah, there's only one, which is like, thank goodness, because you don't have to make an educated guess. All right. So the difference between eukaryotes and prokaryotes in terms of their DNA sequences is that prokaryotes, eukaryotes, have exons and introns. So exon goes exon, intron, exon. So the exons are spaced by introns, and introns are non-coding sequences. Now, in eukaryotes, they have ways of recognizing that intronic sequence and then splicing it out. But we're going to be putting this eukaryotic sequence into a prokaryote, specifically E. coli. And so we need to have those intronic sequences already cut out. And the easiest way to do that is actually just to pull up the message RNA sequence, which is what we got here. All ready for you guys, like a cooking show. And you'll find that often this is already done. Now, there is some extra sequence in here, and I'll show you kind of how to localize that because it'll be important for some of our next steps. So when you want to copy this sequence, you can download the FASTA file, but you can also just highlight this region, copy it, and paste it into a new DNA file. So a new DNA file, for those who are watching, we're actually using SnapGene Viewer. If you want to quickly download SnapGene Viewer, it's absolutely free, and it can be really useful. And I'll explain all this sort of stuff that's going on. So you'll go to new DNA file, you'll paste it in.
So that was the origin sequence you copied?
I copied the whole thing.
Oh, the whole thing.
I copied the whole thing. It says that there might be some non-DNA characters, like the 1, 2, 3, you know, the numbers that were in that area. I'm like, totally remove those. Absolutely. Hold on. Yeah, totally remove those because I don't want them there anyways. Control, copy. I've already done this. I'm just trying to show you guys a little bit more step by step. There it is. Okay, I'm like, all right, this is a linear sequence. It's that long. Perfect. I've already done this, folks. And so when we get to our screen here, look at all these little names. All these are restriction enzymes. I don't think I've really introduced restriction enzymes to you guys yet. We haven't had a lot of reason. We haven't done any cloning yet. This is kind of an introduction to cloning on the theory side. So most of the time, when you get your download your file and put in a new DNA file, it'll have your unique cutters in there. So restriction enzymes are part of prokaryotic systems, so prokaryotes, immune systems. They are endonucleases. That means they cut apart DNA. And these enzymes are there to cut up foreign DNA that comes into the cell on accident or on purpose for the virus. These restriction enzymes made by that prokaryote, there will be no places for that restriction enzyme to bind and cut in the genomic sequence of this organism. So these enzymes, most enzymes, in fact, are very, very specific. And I wanted to show you something. There it is. So the W's are kind of confusing with this one. Let me scroll down a little. That's not so confusing. Okay. So notice these letters. Restriction enzymes tend to bind to palindromic sequences. So if you look, all right. So if you look, I double clicked on it so it doesn't, the letters don't go away. You have AGA and AGA, right? TCT, TCT. It's palindromic. It reads kind of the same from the front and the back. And this is very specific. The enzyme will make a cut, and it will cut down here, cut across, and then cut down. Okay? That's going to leave some sticky ends. Not all enzymes cut to leave sticky ends. Some do something called blunt cutting, and that's going to be straight up and down. Okay? Blunt cuts can match with anything, but they're much harder to put back together. These sticky end cuts, much easier to put back together, and you do this enzymatically. We're also interested in putting our order so that it works. And so we're going to choose two specific and unique cutters. So that was kind of a quick preview of restriction enzymes. Does anybody have any questions about?
It's telling me I need to download the full version.
Just say cancel. All right, and you might not have SnapGene Viewer. You might have just SnapGene.
Yeah, I'm in Snapchat. Yeah.
I'm not the best with IT sort of stuff, so I would just say cancel and then ask for less or try to open a new DNA file. Unfortunately, I don't know the correct answer. Yeah, hi.
So restriction enzymes are some kind of immune system. Wouldn't it make sense that they cut everything that doesn't match a certain sequence? That way it's like, because if it only cuts things that match a certain sequence, what if the invading thing doesn't have the sequence, right?
Question is, wouldn't it be better for restriction enzymes to be less specific in where they cut?
Yeah, more specific about what they cut.
More specific about what they don't cut. Well, these are physical things, first off, and secondly, maybe that's just not a possibility. Maybe that's just not a possibility. There are restriction enzymes that recognize a part of the sequence and then cut to the side. And where they cut, they're not specific to. And those are type 2 restriction enzymes. Those are really helpful, and those are really cool. Because you can put whatever sequences you want in there. And you'll see how that's useful when we start going through this process. So I guess there are some that kind of fit that category, but they're still going to recognize a specific sequence and then do the work. And like I said, that might just be because that's the way it has to be. Any other questions? Cool. All right, so I'm going to go over to our plasmid, BJE202, that we were looking at last time. This is from our previous emails. Please let me know if you need this for doing the homework. Because yeah, you guys will have homework if you want to, which is doing the second gene that's in the notes, all right? Let me simplify this really quickly. So the way I'm changing how many restriction enzymes are shown is I go to the top left corner and there's some scissors. And that's SnapGene's way of just representing restriction enzymes, because they cut. Hey. And I'm just going to reduce it to unique cutters just to simplify our image for a little bit. All right, so we're doing two things. First, we're plopping a new gene into our plasmid vector. This little switcheroo. But secondly, we want to make sure and know that that gene got plopped in. There's a couple ways of doing that, but the way that we're going to do it is by making sure that this gene, our Lux operon, is no longer being made. We're going to disrupt that gene. So that's one way we can know without adding another antibiotic resistance marker. Our next class, we're going to be going over experimental design and some other physical steps, kind of combining the lecture class and also some of the skills that we've learned in the lab class, and kind of putting those together to actually say, like, so actually, how would you do this? Really? How would you know that you got what you got? Normally the Lux operon is not the one, not the gene that you would knock out. You might want to knock out green fluorescent protein, or something that's doesn't have a time-dependent variable with it. Or if this was behind a slightly different promoter. All right, so we're going to be putting our gene somewhere in here. I can't tell you exactly where that's going to be yet, because I haven't told you how this is going to work, right? So what we're going to do is we're going to take two restriction enzymes, and we're going to use them to cut both our plasmid here and also our gerylin diphosphate synthesis. Synthase, sorry, synthase. ACE means enzyme. And the thing is that we want this entire enzyme to stay intact. We don't want it in a bunch of pieces. So we have to make sure that our restriction enzyme cuts this, our PGE202, but not our gerylin diphosphate synthase. All right. Before we get started in understanding about where we're going to cut, we need to know a couple other things. we need to know where this enzyme starts and where it ends. How are we going to figure that out, folks? Does anybody have an idea? No? Something really cool and I think amazing about biology is that pretty much all genes, all proteins start with the same amino acid, methionine. It's not 100% true because it's biology. Nothing's 100% true, but enough to be like, yeah, we just need to look for that codon. That's ATG. Okay. Now, something that's kind of going to help through this process is making a Word document with this edited version of our gene, of our mRNA sequence. It doesn't have the numbers in it anymore. Thank you, SnapGene. And now it'll translate perfectly into a nice Word document. And so I've actually done that in my notes. And you'll see that it's useful a little bit later because with any sort of Word document, you can search, right? And so we're going to be looking for the first ATG, right? And it's going to probably go through my... I did this without any of the other words on this document, but you can see here's one ATG, and here's an additional ATG, okay? That means this front part here, we don't necessarily have to have in our transfer, right? There's a lot of A's here. This actually could be like a promoter sequence, but I'm not 100% positive.
You don't have to worry that it's in like a reading frame. Like, doesn't it have to show up like in every 3? Like, you didn't,
Yeah.
right?
Yeah, but we're not going to assume that this is the beginning or this is the beginning for this class.
Okay.
But looky here, these letters, all these letters represent the amino acid sequence. Okay, so if you just want to take a little bit of time, I don't know what they all stand for, but M stands for methionine, A stands for alanine or something like that. Yeah.
So we're talking, you had several ATGs and you're saying that the first one, so the first and the second is your protein, but why would so we're assuming that the first one is the one of interest and not?
The first one is probably the start, but to confirm, you're going to come back to the NCBI file and you can look at these letters here that I'm pointing to with my cursor. And these letters stand for specific amino acids. So I promise you, if you go just for this first word, maize, you're going to find that's only going to match one of those ATGs, right? And then you'll know for sure. I didn't do that backwards, so we're just going to assume it's the first one. We're not going to disrupt it. But the point is that there's a little bit of junk in the front that we can cut up if we want to, and it won't disrupt our gene. That's the main thing. But there's definitely ways to confirm it for sure. So this is the translation. NCBI also has, I'm assuming that they took some time and actually took the protein sequence and maybe sequenced it as well, because we can totally do that. It's actually a lot easier because proteins look a lot different from each other than amino acids. Amino acids look a lot different from each other than nucleic acids. Okay. All right, so it's nice to have a Word document to work with because we're using SnapGene Viewer. We're not doing SnapGene where you can actually edit your sequence. We can do the editing in a Word document and copy and paste it. So that's why it's we're hacking it, folks, okay? We're hacking it. All right. But yeah, it's important to know where the beginning of it is. And then there are 3 stop codons, which I had to look up. The good news is I did that before the class. And TGA was our last one. Now, any stop codon will stop the synthesis of the protein. So if there's a stop codon anywhere in here, that's going to be it. Okay. So you see there's some still, there's this, so I look for the first one that appears. Okay, and there are 3. Look at, they have a name. Look up codons on the web, and you'll see where they translate, right? Where the nucleic acids translate into codons, and they'll have an area where it's just all stop codons, and you can use those and you can search for all 3. All right, so that's really good to know. And you can annotate our sequence here with that if you like. You can even search in here for those as well. For instance. You can do that, or one of my favorite things you can also do, a little cheat, is go to these arrows. These arrows show open reading frames. I forgot about this. All right. The long one is our gene. Just because we confirm that, right, by going to our sequence. So it's also, we know that we put in the 5' to 3' sequence, not the complement. So this also is a great way of bracketing our sequence. So we kind of know where the beginning and the end is. This is a nice visualization. It's pretty much all we need for choosing our restriction enzymes. So folks, so I'll kind of switch accidentally from nucleic acid to amino acid. I'm not doing that to confuse you. I'm so sorry. I'm human. Okay, sorry about that. All right. So next we're going to be looking for some places to cut in our plasmid. We're going to be doing a little back and forth, and I'm going to show you a little trick that I discovered, I think probably last time, but also this evening. So I found earlier that there weren't really a lot of unique cutters. So I know these are unique cutters because that's what I selected. Thank you to this program. It tells me exactly where they are. That really did the job for us. And so I ended up having to select, we can do some dual cutters. We'll see if our thing is available. It just kind of, we'll slide down. Okay, it does. All right, good. For our gene of interest, our geranyl diphosphate synthase, we're going to choose all of them. Okay, we're going to choose 6+. And then if you come down to the bottom left of your screen, it says find DNA sequence, which is super helpful for finding stuff. But we don't want to find DNA sequences in this part. We actually want to look for an enzyme feature or primer. So I'm going to click on that. And now it'll be easier and reduce the human error from searching through here to see if that enzyme comes in and cuts. Now, everywhere where this, I see that red line, that's going to where the enzyme's going to cut our piece of DNA. So we can't use TAT1. It's gonna cut our gene of interest apart. So what you gotta do is you gotta come back. So it's kind of a little bit of trial and error, right? I'm sure with Snap Gene Viewer, or maybe if you play with this at home, you might find it a simpler and faster way of doing this. It took me about 15 minutes to find my true restriction enzymes that I was going to use. I'm using 2 restriction enzymes, and because they're going to cut in unique places, and it's going to ensure the orientation of my gene in the right direction for the promoter that's present in this plasmid. If I put in the wrong orientation, it's possible that I'm going to that's the one we want. So orientation matters. Back to our circular DNA. All right, so what's really nice about this is it's very visual. And so when I come through, it's actually going to highlight, notice I'm highlighting NHEI. It's also highlighting an NHEI on the other part of the plasmid. So even though I'm showing a bunch of busyness here, it kind of makes it real easy to see. That's why I like using this sort of thing. Because you could go through and like do this all with a Word document and do searching and then like guess where the, you know, oh, I know this restriction enzyme, I have it in my freezer, I'm gonna use it, but it can be a lot longer. So this is making it a little bit easier. It's easier if you actually buy a program, but why? It just takes a little extra time, no big deal. Okay. So what we don't want to go on the other side of this promoter. This is, I think, what's driving this reaction. I'm not 100% positive because this is not my construction. Most of the time, when you get a plasmid, there's going to be a multiple cloning site. The way you can identify that is there's going to be a bunch of unique cutters all in the same area. So it might have been near here. Or it could have been maybe up here. But there's normally like a concentration of cut sites. It's because somebody made this thing. They wanted to be easy for doing a lot of cutting and pasting. And they wanted the beginning of their gene to be right next to the promoter, so they know it's going to be expressed. This one is not that easy, but you'll get kind of the idea. And you can look at more examples of plasmids that are available by going to addgene.com. That's in the notes, okay? So you can kind of explore, look in there, and there's some more other features that later on in the class we'll go over that I think are interesting when we're playing around with addgene. We'll see if we have time. All right. But yeah, we marked this TEP promoter, and so you can use the promoter. It doesn't matter what gene it's associated with. It's just the logic to say, hey, turn this gene on under certain conditions. All right, so I'm gonna go back to my scissors here again, and I'm going to get some more options. All right, so wow, wouldn't it be great if I could just use this one? All right, so you're saying, oh no, it cuts in 2 places, totally fine. We're happy disrupting our genes here. We can cut, basically we can cut out anything from 100 or whatever right after the TEP promoter all the way around basically to, let's say, 10,000. Right, and we actually, I found an enzyme that's a little bit further down the line, but just right before our OMP thing. Anyway, we don't necessarily need the ROP gene. This just maintains the plasmid at a low number, but we can actually have a high copy number of this plasmid. It's totally fine. But anyway, basically you don't want to disrupt any of the logic of your plasmid. So like the ampicillin resistance gene or anything like that. So you have a lot of space that you can just like chop. No big deal. So, all right, I'm like, cool, BSD-DI. All right, BSD is good enough. I'm going to go to my gene of interest, and I'm just going to push, type in BSD. Just paying attention. Yes, it's BSD. Okay, great. And I'm going to search. Oh, bummer. See this blue line? There. Yeah, I don't even need to see it up there. It's fine. I don't need a full screenshot. I know that this is just not going to work out. So you kind of got to go back and forth a little bit. Let's, ooh, wait, well, this unique cutter. That would be so exciting. Damn, I've used that one before. Great. Okay, cool. Bam. High efficient. HI means high efficiency or whatever. Oh no, it doesn't. HI, never mind. HF means high efficiency. Thank goodness. Oh, look at all this information that I get. Great, okay. BAM, HI, let's put a little BAM here. Alright. HI. Next. Oh no! Another bad choice. Alright, so we don't need to do this forever, but you get the idea that you just kind of are going to piece through.
Does it make sense to look at the restriction enzyme that has the longest number of nucleotides, since that seemed to be the highest improbability of a match then? Do you know what I mean? Like they all seem to be like 4 nucleotides, but if you scroll around, or some of them like 8.
They're up to like 4 on each side, so a lot of them are 6.
So it's not
Some
really
are
a good
like-
strategy. Like you should relax one.
They're really specific. The good news is that restriction enzymes are really specific, and if they're not, this program's going to tell you they're not that good. This one's kind of not that great, or it doesn't work as well all the time. And we'll see if we can find one of those for you. That's a great question. I tend to like to look for ones that are small for our next. Because we're going to be doing a little bit of direct immunogenesis, or actually tacking on some extra nucleotides to our gene of interest. And we could do that through cloning. Or we could order this, we would actually just order it that way, right? But we're going to do it the cheap way. So we're imagining that we're going to take a little road trip up to Northern California and go up to the Grand Firs, and we're going to just take one of those leaves off, little pines, smash it up, and then do a little cloning of that gene. We went over some of the tools of like how to clone a specific gene out of an entire genome. We could do that, but or we might have to actually extract the message RNA. This is a eukaryotic, folks, so it's a little more complicated. We're not going to go into those complicated details right now. We're just going to live in dream world right now and focus on what we're doing here. See if we can find one of the ones that is annoying in a sense. So that's a longer one, right? Little surg I. Oh, it tells you how many sides it cuts too. How convenient. I realize that. So informative. It tells you as much information. So this is a blunt cutter, like I was talking about earlier. Those arrows, those black arrows, show where it's going to cut on both of the sequences. So if the arrows are on top of each other, that means it's a blunt cutter, which can be totally useful. Here's a type 2 restriction enzyme, VSMBI, pretty common. And so you notice that the specific sequence that it has written here, the arrows are not even in that sequence. You have some N's. N means any nucleotide. And then we have N4, so this actually makes sticky ends just to, I guess, let's say the right or downstream of this recognition site. I want to point. It's fine. And so N4 means 4 bases down, there's going to be a cut on the complement strands. All right. Well, folks, I did a little work for us before you guys got here, and I found out that Sol, right here, just double check, Sol 1, in fact, that would be embarrassing if it was. Oh, look, no matches found in the query. We have one. Jackpot. That means that we can use this sometime. All right, level, you know, that Word document that we were making comes in handy because we're going to go back. Yeah. And I double clicked on that restriction enzyme and it didn't work this time. Oh, there it is. Okay, it took a little while. I double clicked on the restriction enzyme and it shows me this window. Okay.
So the one you ended up with doesn't cut the gene of interest in half. What if it is present but doesn't splice it? Can you still use it, or just?
Yeah, as long as it's like upstream, so before our methionine, right, or downstream of our encodon. Totally fine to use it. Absolutely. But I kept on finding, going through, and just like cutting right in the center every single time. And cutting in the center every single time. So it was frustrating. I also,
Question from online.
yeah, absolutely.
In SnapGene Viewer, why are some restriction endonuclease names in bold and some are not?
The ones in bold are unique cutters. That means they only cut that sequence one time.
You can look at the camera.
only one time. Are there any other questions from online?
No, I guess that's more.
Awesome.
I got
Yeah.
a quick question. So
Yeah,
if
of
we were
course.
doing this by hand, I would literally search for GTCGAC in that gene of interest, and if it doesn't appear, I'm good? Or am I-
Yeah, I mean, I use SnapGene and just look for sal. Right, so
Oh,
the way
I see.
we're doing it is, I'll review that really quickly, because sometimes it's a little bit harder. So I did control find, just like that sort of, command find if you have an Apple. And SnapGene Viewer allows, do you want to turn back to the thing? So the question again was, how do we know that that cut site is only in our, that doesn't appear in our gene of interest, or where it only appears where we think it's going to be? So I command find or control find. Gives you, the first option was to find DNA sequence, but I scrolled down and I clicked on and checked find enzyme / feature / primer. And apparently it has a shortcut to do that. And I'm just typing in the name of the enzyme. And finding no matches means perfect. Are there any other questions? Okay, great. Yes. Moving on. All right, so I double clicked. I went back to my Plasmid JE202 and I double clicked on that enzyme that I found that works. And it's SnapGene gave me up this other little screen. Yay! Which tells me all about this. NEB is a distributor. New England Biosciences is a common distributor of restriction enzymes. Tells us a little bit about the restriction enzymes, some more information. I am not sure what these methylation sensitivities mean, but it looks like it's not sensitive, so that's really great. And so we can actually highlight this sequence, copy it, and you know what, I'm going to put it in my Word document because it's going to be helpful later. Word document, I actually have mine on Google Hangout, or on Google. And so you'll see why I always copy and paste everything I do. So this is the unadulterated message RNA, right? Here I describe a different vector that I used first. So we're going to just ignore this when I give it the notes. I'll take it out later. But I'm going to describe this sort of information to you, hopefully before we leave tonight. So upstream. So this is a 5' to 3', copy and paste. I tell myself what enzyme it is. And say, in case I need to double check, maybe I didn't get the right nucleotide sequence or something else is going. Oh look, you already know what the downstream is. All right, let's go in there and go. So I just copy and paste. I make notes and make it as informative as possible myself, because I might be a couple months out reviewing this and looking at it, and I want it to make sense to me. Because finding your gene again and all that kind of stuff can actually be really hard. Take a lot of time. Okay, all right, so we're going back to SnapGene. And so here's SAL1. It's got a couple cut sites. So we need to find a restriction enzyme in this area, anywhere downstream of the second SAL1 cut site. I'm just going to save us a little time and tell you that, oh, lo and behold, this one here, the PFOI, just we're going to double check it and make sure that indeed I was correct earlier in my search. PFO, boom. Oh, no matches found. All right, so we have 2 restriction enzymes that are going to work. All right.
What was the second one?
PFOI. PFOI. All right. Again, copy and paste, so you can double click on that again. And if SnapGene treats me right, yes, it'll tell you all the information. Okay? Oh, this one's from Thermo Scientific. Interesting. Cool. That probably means it's just more expensive. It says here, though, note that the PFOI sites might not be compatible. All right? Now, for us, that's not going to inhibit what we're doing because we're going to be adding this cut site to our gene of interest. We're going to be adding it to. So we need to just make sure that we add the correct N here. And the way to figure that out is going down to the sequence. So right now I'm in the map, bottom left-hand corner, I have my cursor. You can actually click on sequence. And let me highlight this really quickly. PFO is highlighted right here in sequence. Oh, look. Thank you, Snapchat, for taking me right on in there. This is the sequence. It looks like we want a C/G. We're just going to copy the 5' to 3' right now. And then when we make our primers, we're going to do it. So we just have a single-stranded gene of interest that we're working with right now. And so we're just going to keep everything from 5' to 3' and then fix it later. All right. So it looks like, indeed, that's a C. Pretty. Oh. What did I write down? I think I just put an A there because I wasn't. I was rushing. Yeah. Oh, it is C. Okay, great. Now it matches. Okay. So, I just made a new copy of my gene of interest and scrolled it down. I put the code for Sal, and I highlighted so I knew that was my restriction enzyme site. You can use any sort of way of noting that to yourself. I put this. Oh, and it looks like I actually highlighted. Oh, the green for go. Oh, so clever. For the actual start, not the enemy. So apparently we could have cut a little bit further down into this gene, but that's fine. Anyways, for simplification, we're just going to put our restriction enzyme site right here. We're just going to write it in. And then you're going to put some quote-unquote random nucleotides to the left of your choosing. Kind of give you some pointers here. When you are making this type of primer, we want 30 nucleotides matching the template and about 15 nucleotides on the overhang, not including these. So I'm going to make it a little bit short here. I'm glad I put these pointers in and just didn't fall.
Is that just to stabilize the ends of the primer?
Yeah. And also restriction enzymes like a recognition area and something additional to kind of like hang out with.
Sure.
So another way you could have done this is just simply change this a little bit. Boom. All right, I'm just moving my cut site to actually follow the rules that I placed at in here. So you want 30 nucleotides matching on the template and about at least a 15 nucleotide overhang. Okay. Here is, side this with C. Here's, I put the restriction enzyme cut site again for PFO. And all right. The reason why I made a new, I just like to show all my steps really explicitly. This is like my lab notebook. So I like to show everything right down, everything that I've done. We're going to make a new DNA file so we can plan out our primers. So I went ahead and copied this and made a, and this one's not going to be the same. Yep. Wait for it, there it is. All right, because otherwise you're gonna have to like, because you know, the primers you first make aren't gonna be the greatest. There's gonna be some weird complementarity. They're not gonna be maybe the right TM, the melting temperature. Soundgene's gonna help just kind of blaze through that process. Don't you worry. Look, hey look, here's our Sal I, right where we placed it, and here's our PFO. And these are all unique cutters. So this just verifies again that we did the right thing, that, yeah, here it is, PFO. It's like, where did it go? Oh gosh. That indeed, these only cut the sequence once. All right, so that's really important. This is just double checking your work. All right, now let's go down to the sequence level. We'll go to Sal 1, all right? And so the idea, again, you're gonna want like 30 base pairs overhang and like 15 on the other side. And so most primers are about 60 nucleotides long. We're gonna go for about a degree of 60, right? So make sure that you have those sort of ideas. There's a couple other pointers. You want GCs, definitely at the 5' end. 3' end is also good too. Just as, so maybe we'll include one more C here. It's nice that Snapgene also kept our formatting here for capitalization. I didn't know they did that, and it's really helpful. Thank you, Snapgene. And, you know, again, we're gonna go here, we're gonna copy top strand, and we're gonna go check for complementarity. We don't want it. We don't want self-complementarity. We don't 5' or 3' complementarity. And the website that we go to, I have it in my notes here, and okay, it's from Northwestern University, so you can just put oligocalc and Northwestern, and you should probably be able to get to this. But
This is a check for that hairpin like binding and self-binding.
yeah,
that sort of
all
thing.
the annoying stuff that tends to happen. I need to recopy my primer that I made. Okay, control copy. And I put actually notes in this lecture, notes in the lecture notes about what you're looking for using oligocalc. I did not do that in the last lecture because I got ahead of myself, and so they were not included. All right, check for self-complementarity. Yay! Oh no, potential hairpin formation. And so the good news is that there's no self-potential annealing and there's no self-complementarity. So we'll need to redraw this again. Redraw it as in copy, paste. So I'll just show you how I kind of normally navigate around. Sorry. So, all right, I'm like, okay, that didn't work. You know, I can shift a little bit. So on the left-hand side might not be as important. And so I'll just shift over a couple. Oh, I said 5' is better for the C's and G's. All right, we'll shift over this way. And again, we're just going to go for about 60 degrees Celsius for our melting temp. Okay. Going back to our oligocalc. Okay. Yay! So just shifting over a little bit can actually have a huge effect on what happens. And when you see none, exclamation point, none, exclamation point, none, exclamation point, you can celebrate like I just did.
So you didn't change it and you just looked in a separate place?
I shifted it over to nucleotides. So I highlighted 2 nucleotides upstream, and then again, when I highlighted, it tells me the melting point. We want something about 60 degrees Celsius. Okay.
And how did you know that you wanted that melting point?
Because that's what you do for polymerase chain reaction, for the physical way of making more copies of a gene. Great question.
What does it mean for the DNA to melt?
Great question. So what does it mean for the DNA to melt? DNA is double-stranded. Okay.
Oh, is that where they separate?
And so that's when they separate. And you can actually tell this with light. They would shine light and the absorbance changes. And so genomic DNA completely melts at about 98 degrees centigrade. And then it's based on the number of nucleotides that are binding. Because you're breaking hydrogen bonds. They're not covalent bonds, but they do have some energetics holding them together, and you have to get them moving enough so that they're like, okay. That's what happens. Cool, I didn't know we were going to find this so quickly. It makes me super excited.
So that means that you know air information.
Yeah. According to this algorithm, and so that's really, we're just going to trust it. We'll copy. And we're going to guess what? We're going to put that into our notes.
So is that just checking to make sure that the DNA probably won't like stick on itself and stuff like that?
Yeah, it won't stick on itself. I'm actually going to copy it because of the formatting from our SnapGene file, so I don't have to remember where our restriction enzyme cut spot is. Although you can find it pretty easily because it's unique. Wrong one. I clicked on the wrong SnapGene file. It's good to label and name your SnapGene files, especially when you start playing around with cloning. It's a nice record of the steps that you took, and then you don't get them confused. Yeah. So. And then I just go back to my Word document. And then just for my own reference, we're going to highlight those. Okay. If we have it at, if we had made our primer at the tail end of the gene, we're going to want to make sure that our overhang covers for about at least 15 nucleotides. The rest needs to be 30 nucleotides, like I said, as a primer, need to be on our template strand, and the template strand means on our messenger RNA,
So
our gene of interest.
So what is that overhang of 15 nucleotides? What does that do? Is it?
It allows for to extend the sequence further because you want an extra overhang because the restriction enzyme doesn't do well if it's the first nucleotide it recognizes is at the end of the sequence. You can imagine that's the part that's moving around the most. Yeah, it's like whipping around.
Right.
And so it gives a little bit more for it to grasp onto, and you just have higher efficiency that way. I'm just giving you a possible reason why, but you have higher efficiency that way of cutting. So it cuts more of your pieces more thoroughly. Okay. All right. And so I'm going to go back to my map. I'm going to click on my PFOI. I'm going to click back onto sequence. It just localizes me really quickly and easily to the area that I want to be in. Same sort of deal. I look some G's and C's. Perfect. That's great. My 5' and what if there's some that are really close in? Yeah, we'll use those guys. 64. Your primers need to be within 5 degrees Celsius of each other. So they can have different melting temperatures. Sometimes it just happens organically because there's more cytosines and guanines. Those impart a higher melting temperature because they make 3 hydrogen bonds rather than 2 with their complement strand. And so they hold just a little bit more tightly, bringing up that melting temperature.
So you said it should be similar, which is 5 degrees.
Yeah, they should be close. Although I've seen papers where they're like 10 degrees apart, and I'm like, what am I going to do with my PCR program? And this was actually a paper, really? And so I struggled, and there's some ways to get around that. That we'll probably talk a little bit about in next week's lab on Sunday, 10 a.m. I'll probably still share the notes for that lab. So if you're unable to make it, I still need to share the notes from yesterday's lab. And if you're here, I also reran and got all positive for people who left their samples with me. Everybody had plasmid in their samples of different amounts. There can be reasons for that, but anyway, I'll send out that email. If you remember your number, you're good to go. If you didn't leave your stuff with me, I'm sorry. You can just believe that I trained everybody well enough that everybody got a sample. I'll make an announcement on the email about that sort of stuff. Anyways, all right, control. Happy, but actually we're gonna go times 2. What if that really matters? I'm gonna have to do. I'm gonna do a little bit. We're gonna do control. We're just gonna copy this. So I don't think it actually. Oh, we're gonna go back to our oligo calc. You need her. Right here. All right, I'm actually gonna test something out. I'm not really sure. So before I tell you guys anything, I'm going to. Oh my gosh, this is like the worst site ever. Self-annealing isn't the best. You imagine that the melting temp is going to be higher than this, but this is actually a pretty good amount of self-annealing. And what this does is that it's much easier sometimes for the primers to attach to themselves than attach to your template, and so you end up getting a bunch of junk. And that junk builds and builds and builds on itself, and you get a false band, and just reduces your efficiency of your PCR reaction. I'll do that little shifto changeo. Yeah.
Is there any way to predict that kind of thing without just trial and error?
Yeah, using an oligo count.
Well, yeah, like trial and error.
And you could also just try it, of course.
Okay.
Of course, but oligos are not free. They cost. They're not that expensive though, so they don't work. They're like $8. You know what I mean? It's not a big deal. But
for like the hairpin part, you're just looking at a complement. Like is that complement?
it's actually self-complementing.
Yeah,
it's making.
self-complementing.
It's doing this.
You can write it to it. Like that's what they did, right? They wrote a program, so
that you wouldn't have that same unfortunate thing happen to you over and over and over again. You could have an idea that your primers would work better. Okay. All right, I'm gonna try one more, and I'm gonna actually try. I'm curious. This is even worse. Oh my god. You can also make them longer. I'm not gonna bore us too much longer with this. You have an idea about how to make primers, and so I'm just gonna try one more. T's and C's on both sides. Good. To our oligo calculator. All right, this one you just gotta play around with it some more, and figure out the thing that has the least amount of stuff. Now, you want to minimize the number of self-annealing. So if it's like 3 or something like that, your melting temp for these guys will help with that. So there's ways of calculating exactly how sticky this is going to be. You want to just reduce the number of hybridizations like this and hairpins, because there's actually a temperature where this hairpin formation will happen most likely as you raise the temperatures. Both this and this will go away. So play around with this at home. We don't need to spend too much time. We went over primers a little bit last class. All right. I wanted to spend some time going over using vectors using AdGene, but I wanted to open up the class to some questions from our last session and questions from this session. Anybody? Yeah.
Yeah, I'm still a little stumped about the things you were highlighting. So If you did one and it didn't work out, but then you moved it over, and then once you find one that works, you're just gonna save that section and that.
Yeah, I'm just going to copy that and put it into my notes.
And so those aren't as particular. You don't need like a certain start code or an end code for those. You can do those randomly.
Right, you're just looking for a certain melting temperature and reducing you want to see it read none, none, and none. Basically. None, none, and none. And then you know that you're good to go. One last thing I forgot, and you remind me about this. Thank you, somehow. When you order your primers, notice I copied the 5' to 3'. When you order your primers, you're going to want to go to your SnapGene viewer, and you can find the sequence again, right? You have it in your notes on your Word document. You go here, let's say, okay, we'll copy. And then you bring up your find, right? And I just say I copied this from my Word document. I put it into the find area. I'm like, next, please find this. Oh, there it is, yay, okay. There's the non-working primer, a sequence that we're totally not going to use, but I'm going to use it as a teaching moment. When you order your primers, folks, this is in the notes, but you want to copy the bottom strand. Okay, you want to copy the bottom strand from 5' to 3'.
The default is top.
The default is top. Top is always going to be 5' to 3'. It's never going to ask you. 5' to 3' is going to do the bottom strand in the orientation from the right to the left. I have the double T. Oh, it tells you. Yeah, 5' to 3'. So you want it's reading it backwards, and it tells you this by the CTG, right? And it's like, do you not know what 5' to 3' is? Don't you worry, we're going to remind you. And you're like, oh yeah, I want it going from the right to the left. Okay, not the left to the right. Okay. There is a reason why you would do it the opposite way. And you can always decode it, folks. You know how to match these pairs. It's A to T, T to A, C to G, G to C. So it's not too hard to figure out. But that's what's available. And yeah, I just highlight it, and when I highlight this sequence, notice there's a purple bar here on the right-hand side that's showing my TM. And you have to keep the button click on for the TM to continue to be shown. TM means temperature melting. And this is 63 degrees centigrade. Yeah, you want your primers to be within 45 degrees C up to no higher than 70 degrees C. Okay, so there is some range. The longer the primer is, the more specific it's going to be to your sequence. But the more chance that it could have some self-complementarity to it, and where you can get some what's called primer dimers, which is an artifact in your PCR. Okay. So I want to show you guys Agene, where a lot of people get their plasmids from. If you don't have a plasmid, here's my roommate. You don't have a plasmid already. I like to sometimes get plasmids from anybody if I can. But I can get it full free. It's even better. That's why we're probably going to be using the PG202. Okay, even though it's not the most convenient one. But there's also, like, if you ever, for some reason, buy, or somebody else buys competent cells, they normally have a control. So one that's like, this one should work, and you get this number of colonies when you use this number of microliters in your reaction. Hey, that's still a plasmid. Hey, it still has a multiple cloning site. It has a selective marker on it, and can be really helpful. But my favorite ones, my favorite what's called vector or favorite backbones have antibiotic resistance marker and a differential marker. So they might code for beta galactosidase, which you can put something in the media, and when that gene is working, it makes your colony blue. When it's been disrupted, your colony is white. So it's a differential marker. Okay, look at that gene. There's a couple other things that we can look at as well. This is why I kept in my notes which vector I used earlier. That is. I'll show you how. Okay, that's great. All right, wonderful. Add gene. Where are we? I guess it is in my search. Great. Let's go to the home page. Now, Add gene actually has a lot of just great basic information about primers. I even cited those in some of my notes. And you can search for plasmids. You're like, oh, I want to look for E. coli plasmids. All right, so let's look what Add tells us about the plasmid. No maps are available, that's fine. Normally you're going to get some or all of the sequence. Pretty much only want to buy plasmids where you get the entire sequence. All right, so I was giving you a source. So you can search by what is this? It's going to find plasmids.
So again, how do you insert that?
Sure. There are a couple ways. One of the ways is a chemical transformation. That's one of the most common ways. Not all organisms can work like this, but a chemical transformation basically puts your E. coli in an environment that, all right, it puts, first you put your E. coli with some divalent cations. It's divalent cations are positively charged. What they do is they shield. So they kind of get around the really negatively charged DNA, and they kind of get around the really negatively charged bacterial membrane, and they kind of prevent them from like super interacting and bouncing off each other because they're like soften the charges. And then what you do to this media is you got your DNA in there that's from the outside. It's called exogenous DNA, right? And you have your cells, and you have them in this chemical mixture. You heat them for like 30 seconds. You heat them, and that loosens up the microbial membrane, allowing these pores to happen and allowing the DNA to slide inside.
Plasmid.
Yeah, plasmids or even single-stranded DNA. Then you put them on ice again, and it closes up those pores. And then some live, some die, and you put them on some selective media. Instead of a heat shock, you can electrocute your cells, and that actually works more faithfully across more organisms. Another way you can get your genes in an organism is actually to shoot them in using a gene gun. So that's where you take this basically fine particles of either tungsten or gold, and you coat them in DNA, and then often some sort of protein coat, and then you just like launch them into your tissue. And some cells live, some cells die, and through homologous recombination or something along those lines, they can take up the DNA.
Well, plasmids can be transmitted between different species of organisms, right?
Yeah, in the bacteria,
Viral
absolutely.
or whatever.
Yeah, so we're not quite done with our list.
Right, great.
There's also in vitro fertilization, right? And then lastly, another natural, there's two other natural methods. One is, I'm forgetting the name right now, but basically using a virus. And so using the virus machinery, which is the protein, to insert the gene. And then lastly, what you're talking about is basically sexual reproduction with bacteria. And they actually have to have an accessory bit of protein to make a sex pili that can connect with another bacteria to share that DNA. And the way you do that in the lab is you're like, all right, you're the one with the DNA for the sex pili, you have another one with the gene of interest, they're sharing, and you plop it on top of the one that you want to just take the gene. something along those. So it's normally like 3 different organisms. With their sex pili, that's basically a quorum sensing molecule that says, come hither. Yeah, so I went to browse. I went find Plasmid's browse because I'm just actually interested in some features. So here, some really cool features. Oh wow, these are really popular. They're actually on fire. CRISPR, so cool. Oh my goodness. Million. AI, let's interfere with some genes. Empty backbones. That means there's not really anything going on there except, when I say going on there, there's not any genes to code for anything else except for, hey, this has this origin. So it's not really a gene, but the logic for saying, hey, replicate this DNA, make more copies of it. And, oh yeah, make this number. Oh yeah, and make sure that all the daughter cells get the same number of plasmid. There's a couple other genes that people have inserted into these backbones to make science easier. Because you could extract plasmids randomly from the environment, sequence them and use them. Optogenetics is light sensitive primers on plasmids. Wow, stem cells, what? Okay. You can also look and see where, when it says expression system, that means when you put a gene in there, it's going to be made in that organism. Replicate it in another organism, like plants or an animal or something like that. This one is empty backbone. So they removed erythromycin, so that's why it's one of the empty backbones, I suppose. It grows in DH10B, which is a specific type of E. coli. Specific strain, so you can keep that in mind. This is a low copy number. So the difference between a low copy number and a high copy number plasmid is copies each time. That's how much E. coli will keep per cell. High copy numbers, we're talking about 300 or 500 copies. For expression systems, for ones that are going to be making a protein, you probably want like a low copy number. You don't want to stress out the cell to make tons of DNA. But if you're using E. coli just to make a bunch of really cheap copies that are pretty faithful of your plasmid to do other downstream things, yeah, let's knock it up a bunch and put it to the 500 or 300, high expression. It tells you what this plasmid is resistant to, so chloramphenicol. That's good to know. Sometimes they'll tell you more information, but I want to, before we close out, because it is getting late, I'm just going to, feel free to browse this. Like I said, the link is on the notes. Oh, wrong one. Okay. Here's the plasmid that we were going to use, but I wanted to work with something that we were familiar with already, so it was less intimidating. I wanted to use a plasmid that you guys were familiar with. This one I liked because it has the LAC-Z gene. So gene or insert, right? LAC-Z. This is LAC-Z codes for beta-galactosidase. So you can do blue-white selection with this plasmid. So you can knock out the LAC-Z gene, interrupt it any part, the promoter area, probably keep the promoter, any part of the gene. If you cut out any of it, it's not going to work anymore. Just not going to work. And then you'll be more confident that your new gene that has been put in correctly. It may not be expressing, but you know they've inserted the DNA into the plasmid. This happened to be a high copy number. It grows in DH5 alpha. It will probably work in all E. coli, all right? But it'll probably be sent to you in DH5 alpha. It's like multiple cloning site, which is, or this is the cloning site probably for the laxine. I just need to take a little bit of time to go into this more deeply. But the things I took home from this information was that it's gentamicin resistant, it's lax-C, and it's a high copy number. Growth temperature, pretty much, E. coli loves 37C. It grows in people's colons, it grows in animals' colons. 37C is human body temperature.
Hey Mary, I have a question. What plasmid are you looking at?
This plasmid right now is PRU1103.
So how does the computer algorithm calculate the temperature that needs to be built?
There are several different methods, but it used to be just experimental, right? So you have, you could guess or get a pretty good idea.
Actually heating the thing up and watch it.
Yeah, so first you would separate, you would use a restriction enzyme, and you would just cut up a bunch of DNA, like a restriction enzyme, and then you would separate those on a gel, and then you take the smaller ones, you do gel electrophoresis. You take some smaller bits of the DNA, and then larger ones, and then you look to see where the absorbance changes. And actually you can see, like, I think it might be visible if I remember correctly from the stories, that like how the media looks changes. I don't know if our eyes are that sensitive, I can't quite remember. But you can definitely use a spectrophotometer to tell the difference. The absorbance will change for a certain wavelength of light.
Well, it means like it's a matter of the chemical composition.
Yeah, exactly. So there's that. Then they're like, all right, cool. We will look to see based on the number of guanines and cytosines that are present. So once we were able to start sequencing, we had a better idea of what was contributing. And so the number of nucleotides that give a 3 hydrogen bond, so that's cytosine guanine, right? Versus adenine and thymine, those are 2, sorry. They get 2 hydrogen bonds. You can make a pretty good guess based on the composition of triple hydrogen bonds versus double hydrogen bonds about what the melting temperature is going to be. It's a great question. Anyway, that's all I'm going to talk with you today. Unless you want more questions answered, I'm here.
All right, I'm gonna give the online people one couple minutes to ask questions.
Oh yeah, it's totally fine. Yes.
So what is silico plasmid? What is that?
That's just a fun way of saying is that we're doing it on the computer.
Oh, okay. So
And so instead of like, because they have like in vivo and in vitro, so in vivo is in life, and then in vitro is like without cells, and in glass, because vitro means glass. Yeah. And yeah, so it's kind of like a play. They're like, oh, well, yeah, in silico. It attracts all the IT people.
If we're waiting on another question, from online,
Yeah.
can I ask just real
Of
quick?
course.
So when we're looking at the sequence, and it shows if it were to be able to express this restriction enzyme, it would be inside that circle, right? And because there's a differentiation between that plasmid being able to express the enzyme versus being able to get cut by the enzyme. So if it's that dash outside, it means I can be cut here. But if it were inside the circle, it means I can generate this enzyme.
Yeah,
right?
you would, let's see, let's go look at a picture. All right, so if it were making the enzyme,
It would be inside.
it'd also be a bunch of nucleotides, and it would be in an open and reading frame. This is just pointing to a 1
one
Points by.
specific, I got it.
Yeah, that's good though. That's really helpful. I did not make that clear.
All right, so the video is going to be up on the link I gave everyone, and we're going to leave it up there for probably like a week or so, and then we're going to probably take it down. But
It's unedited.
Rough crowd.
but you guys are welcome to review.
I have another question.
Yeah.
Yeah.
I'm a loser and I arrived late. What late?
I emailed it to everyone who's been signing the things who've been
That's
passing
okay.
around.
He didn't get it because his first class was last lab.
Oh, okay.
So I guess I should take that link and.
You should
Did you
take-
also put it on the meetup?
I did. You should also make sure that you add your name and email to that sheet.
Well, this sheet is. I mean, we can do this one as well. This is for the lab too. I think you had it on there, but anybody who wants who's new and wants notes from this class, please put your email down. I also like having your emails down for each class because then it's like taking attendance, and it's easier for me to sign off that you're certified.
Can you put like a... Oh, okay, that's another sheet.
Yeah, I just put on the back. Would you mind biohacking lecture 4?
I'm trying to track who goes to what lab, so that if we do give people certifications, we can kind of track their skills.
Yeah.
So for all of this is the notes and the video, and that's all linked to in that.
The notes I send out after the class.
Okay.
You haven't had the opportunity to appreciate my lovely follow-up emails that invite you to the next class, as well as give you some additional information, clear up some things that I thought were confusing, and link to a Google Docs which has my working notes that I use for the lecture. And I think they're fairly thorough. I mean, anybody else can say that they're not, and that's fine. I'd love to add to them more. But yeah, I try to make them fairly
Hi, everybody.
more and more thorough as we go through. The good news is also if you
Well, they
missed
can't see me here.
previous lectures, I linked to all the previous lecture notes.
So this project,